he's going to talk about bacterial motion, basic physics, geometry, and biology.
Okay, thank you. And, oh, Jair, very good. Okay, so my talk will be much lighter than anything else you've seen. I have lots of movies and only a couple of equations. And I just threw those in at the last minute, actually, to sort of justify doing this talk at a geometric mechanics conference. So they're sort of artificially inserted into the middle of my talk. Okay, and yeah. So my main collaborator on this over the last 20 years has, of course, been Jair, and I put some of my other collaborators there. And I sort of extracted this talk from a talk that I gave to my students. And I wanted to show them sort of the interdisciplinary nature of the subject. So we have a nuclear engineer in our in the group. We have biologists. We have an MD. John Heuser is actually an MD. We have some physicists, and we even have a philosopher, who now I believe works at the planetarium. Yeah, the head of the planetarium. Yeah, I remember at one point Jair torturing the guy because he said he wanted to become a philosopher, and Jair made him get a master's degree in math first, and made him do some hard analysis or something, and before he was allowed to pursue this. This is a wonderful picture of Jair here before we almost lost our lives about 20 years ago. We had a wonderful trip across the bay, and then a huge storm started on the way back. I don't know if you remember this, but we came back, I don't know, 4 hours late, and we had some children with us, and their parents were just out of their minds. But so adventures with Jair. Okay, so here's the questions that I address in this line of research. So what is life like for a microorganism that swims in water? How do they move? Why do they move? How do they sense their environment? And how do they control their motions to move to more favorable regions? Okay. Well, these questions were addressed in this very famous paper by Purcell. And this was 45 years ago. He wrote this wonderful paper called Life at Low Reynolds Number. Okay? And it was one of the inspiring papers for us. Okay. Well, we got our start about 20 years ago when we had a meeting here in Rio, organized by Jair. And Jair invited Howard Berg, who's one of the leaders in this field. And we wrote a paper that came out of this meeting called Problems in Progress in Microswimming, and that sort of laid out our program that we followed for the last 20 years. Okay? And we've expanded on this greatly. We've added many more problems. Not a lot of progress, but we've added many, many problems. That's probably... And here's a quote I took from one of Howard Berg's books. And he calls this a modeler's era. We're entering a new phase in the study of chemotaxis, in which enough is known about the detailed properties of signaling network that its behavior can be treated analytically and simulated numerically. It's becoming a subject more in tune with the physical sciences, with the constructive interplay between theory and experiment. That's a quote out of one of his very wonderful, very thin, to-the-point books that he tends to write. Okay? So swimming microorganisms live in a very unfamiliar world. And I just put these numbers up here to impress my students with great numbers. Okay? So we live on land, and we're typically about 75,000 grams. E. coli live in water and have a mass of, well, whatever number that is. And so we are that many times more massive than an E. coli. And that changes the physics quite considerably. Okay? So in the shoes of a bacterium, what's life like if we were a bacterium? Okay? Well, First, if we try to move, the water sticks to us. The drag is overwhelming. I'll explain this a little bit more in a bit. It's almost like swimming in tar. But on the other hand, we're subject to Brownian motion. Okay? We're just constantly being jiggled around and knocked off course. And that has very profound implications on how we control our emotions to get from one point to another. Okay? And then to collect nutrients. The way we eat is we actually wait for molecules to diffuse through the fluid and find locations on our cell wall that are chemoreceptors. And then we capture the molecule. You can't just simply swim up to food and eat it like a fish or we might do. Okay? Why swim? Well, bacteria swim to improve their situation. Some swim to escape chemicals in their environment. For instance, sulfur-reducing bacteria are oxygen phobic. And I put a little note to myself. One of the wonderful things that we saw. Oh. Jair told me to mention that they also swim for sex. So. Anyways. We saw some, in one of the labs here in Rio, we got to look in a microscope and look at these wonderful bacteria that are magnetotactic. And what they did is they had a microscope, and they put a little magnet next to it. And these bacteria have a little compass inside. And they find their way through the fluid by looking at the magnetic field. And they always swim along the lines of magnetic flux. Okay? And you'll find these bacteria here in the lagoa, believe it or not. Okay? I guess evidently they're very common in the lagoa. And what they like to do, they don't like oxygen. And they try to swim to the bottom of the lagoa to get away from oxygen. Okay? And it turns out if you swim north, if you're in the southern hemisphere, if you follow the Earth's magnetic flux lines, that takes you down into the mud where they like to be. And it's a wonderful experiment you could do. You could take these bacteria up to the United States, and in about 24 hours, guess what you have? You have south-swimming bacteria. The mutations build up so fast that in about a day, you actually get bacteria that swim to the south. Okay? There's one way that bacteria control their motions, is using little internal compasses, little ferrous crystals inside of the bacterium themselves. Okay? Some are attracted to certain chemicals, and E. coli, for instance, is attracted to sugars and proteins. Okay? One of the organisms that Jair and I have studied for many years, Synechococcus, this is sort of still an open question. We have some ideas about why they swim, but for many bacteria, we just don't know why they swim. Okay? So, self-propulsion of a bacterium through a fluid. And this is a quote from an actual bacterium to Purcell. And it says, the bacterium said, "What is this thing you call inertia?" Okay? Well, Reynolds number. Okay? So for fluids, the Reynolds number is based on size, speed, and viscosity of the fluid. And it measures the relative effects of inertia and viscosity. Okay? I.e., the ability of a swimmer to coast. Okay? So human swimmers are able to glide a great distance between swimming strokes. I can remember when I was in college, I went to the United States Naval Academy, and we had to learn to swim, obviously. And we had to be able to get across the pool, I think, in 6 strokes. And of course, the swimming instructor, he was this big barrel of a guy. And he'd stand on the edge of the pool, and he'd totally inflate his chest. And he'd dive off, and in one breaststroke, he'd end up on the other side of the thing. And of course, the rest of us would jump in and pop, 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 pop. It'd take us 20, and we'd fail and have to go to remedial swimming lessons or whatever it was. But humans can coast quite a long distance between swimming strokes. Ships, for instance, can take miles to coast after their engines are shut off. This is actually a ship that I was on. I lived on that ship for about 4 years. Okay? So I have some experience with this. Okay? When the Reynolds number is much less than 1, viscosity dominates over inertia. And here's one of Purcell's famous back-of-the-envelope calculations. He asked, what if you take an E. coli that's swimming at 10 body lengths per second, and then it just suddenly shuts off its engines? Pow! How far does it coast? Okay? So he just wrote down Newton's second law, where he used the force the force required to translate a sphere through a fluid, right, at this speed. Okay? And matched that with mass times acceleration and computed. And here's the result. The E. coli would coast for about 0.04 angstroms. Okay? So it coasts a distance small compared to the diameter of a hydrogen atom. Okay? Okay? So inertia plays very, very little role in the lives of E. coli. So the quote from Purcell's paper is that F may equal ma, but E. coli could care less. Okay? And in a sense, microorganisms follow Aristotelian physics, right? No force, no motion. Okay? Velocity is proportional to force for an E. coli. Okay? Okay? Well, does any of this have anything to do with humans? Well, over the summer, did I send you this article from Scientific American? Not this one. So this is a picture out of August Scientific American, and it describes some of the things I'm talking about. And it had this picture from the Boston molasses disaster of 1919. Okay, when a 5-story vat of syrup split and flooded the streets of Boston. Okay, and 2 million gallons of syrup poured onto the streets, and a 25-foot wave of syrup came down the street, right, carrying people along. Okay, well, evidently, it destroyed buildings, killed 21 people, and evidently one firefighter even became exhausted trying to swim at low Reynolds number, and the word is that he actually drowned. Okay? Well, taking into account the difference in scale, swimming bacteria must overcome viscous forces more than 1 million times greater than those suffered by the Boston rescuers. Okay? Yet they do pretty well. They swim pretty well. Okay? So, like I said, I threw in a few slides with some formulas on it, with some equations on it, and here's one of them. Okay? So, equations of motion. So, we start with the Navier-Stokes equations, and of course, we set the force term equal to 0, and this is called the Stokes equation. And we apply no-slip boundary conditions, so the fluid velocity adjacent to the cell wall matches the fluid velocity adjacent to the cell wall matches the surface velocity. So it's a deforming membrane. Okay, and the nearby fluid matches the velocities match. Okay, and of course the fluid velocity is decayed to 0 away from the cell. And James Lighthill, one of the pioneers in this field, says this is really fluid kinematics, and not fluid dynamics. Okay? Okay. Well, key observation. The net force exerted on the fluid by the organism is 0 at each instant. Fluid motions should be reversible. And here's a wonderful demonstration. And if I were an experimentalist, I'd do this experiment for you. When we had our meeting back 20 years ago, Howard Berg actually brought a little vat of glycerin and did this experiment. And I could never get the thing to work. So I found a movie of this experiment done by G.I. Taylor, one of the big names in fluid mechanics. And I'll show this. Okay? So what he's going to do is he's just going to put a little drop of dye into a fluid and then mix it. Okay? no video tricks here. Okay? This is old. This is from the 50s, right? What's that?
The thing is the trick there. You see this extra finger there?
Jair said it's the extra finger, the white finger, that's doing the trick. And there you go. This is probably glycerin. I would imagine it's glycerin. So you could stir a low Reynolds number fluid, and then if you very carefully retrace your steps, you could unstir the fluid. So he took some dye, mixed it into the fluid, and then very carefully unmixed it, and you get the little blob of dye back. Okay? Well, Ludwig Wilhelm in the 1930s was the first to realize that reversibility has an important consequence for low Reynolds number swimming. Reciprocal motions cause no net motion through the fluid. Everything is reversible, right? So if you have a little scallop, right, and it tries to swim by just opening and closing the hinge, what it'll do is just oscillate back and forth and get nowhere. Okay? So this works at high Reynolds number, right? You could snap the thing closed, right? Emit a jet of fluid out the back, and inertia carries you along, and opens slowly and so forth through the fluid. And this doesn't work at all at low Reynolds number. So you just oscillate back and forth. Okay? So microorganisms have to adopt strategies that don't rely on reciprocal motions for progress. Okay? So I guess if you happen to encounter a molasses spill, if you're visiting Boston or something and a vat breaks, don't just flutter kick your legs. Okay? You're just not going to get anywhere. So you need to use maybe a breaststroke. Okay? You can't use reciprocal motions. So if you have something to take home today that might be useful at some point in your life, there it is. If you ever encounter a molasses spill, you know what to do. Okay? A couple of nature's solutions. Okay? Well, many bacteria, have what are called prokaryotic flagella. These are actually little propellers that spin using little electric motors. Okay? Some organisms have internal flagella. Okay? This is a bacterium called spirochete up here in the upper right. And maybe in school, you've looked under a microscope and seen paramecia. And what they have are little hairs called cilia, and they stroke in a coordinated fashion, and you get these rows of waves passing down the organism. And all of those work just fine at low Reynolds number. Okay? Well, here's a sketch of the basic model that got Jair and I interested in this problem in the first place. And this is probably recognizable to lots of us. But we think of swimming at low Reynolds number as being holonomy of some connection. Okay? So down in the bottom there, I have, well, that two-dimensional space there is typically infinite dimensional, but I cut it down just a little bit. And down below, I have my shape variables. Okay? And those are the controls that my swimmer has. So a little loop in there would represent a swimming shape. And then what I want to do is, using the no net force and torque condition, lift that curve and figure out what the net motion through the fluid is. Okay? And this idea actually defines a connection on what's called a SE3 fiber bundle. Okay? So to us, swimming is actually holonomy. And I guess I have we like to think of the lift of this curve as being a horizontal lift. And these little planes that I've drawn are the horizontal directions. Those are the motions that not only encapsulate the swimming, a small piece of a swimming stroke, but also the infinitesimal motion through the fluid that's necessary to cancel the forces and torque on the fluid. Okay, and these describe a distribution, and I drew two pictures here. So I drew a picture of what would be maybe a pinned organism. Okay, and if I pin him down, he'll actually exert net forces on the fluid. And I sort of indicated that by drawing a little plane up here, right? And the velocity vector of the lifted curve is not tangent. Okay, and that indicates that we're not meeting the no net force and torque condition. Okay, but the free organism, the upper one, you'll see that the velocity vectors are all tangent to these planes. Okay, just a little bit. So how do we compute force and torque? Well, the first thing that we needed to get is this thing called the stress tensor. Okay, and we're actually able to we compute it using these formulas right here, and this is the stress tensor, and what it gives it's a matrix, and sigma ij. So this entry is the ith component of the force per unit area in the jth direction. Okay, exerted on the cell by the surrounding fluid. Okay. Well, in some of our papers, we have something called the power metric. And I said here that the swimming connection is actually a mechanical connection. Okay? So the power expenditure exerted by the cell in deforming from a shape, so I'm calling sigma a shape, right? And if it moves just infinitesimally, right, some work is done, and it's given by this expression right here. Okay, and we could form an associated bilinear form that looks just like this. And there's a very important theorem in low Reynolds number fluid mechanics called the Lorentz reciprocal theorem. Okay, and it tells us that I could switch u and v, and I get precisely the same number. Okay, so this bilinear form is actually symmetric. And I believe it was Lorentz that showed that it's also non-degenerate. So we could think of this as some sort of a Riemannian metric. Okay? And here's a nice fact that I think Jair actually found first. It was we actually rediscovered it over, I don't know, maybe a 5-year period. But I think it's actually in our first paper, Problems in Progress, that the horizontal subspaces that I mentioned earlier are perpendicular to the vertical subspaces of this in this bundle. Okay, so we actually have a mechanical connection. So here are some formulas for estimating the swimming velocity. And let me just shoot right through these if you don't mind, because I have many wonderful movies to show you. He said I only have 5 minutes. Okay. Well, the best studied organism is something called E. coli, and this is an organism that lives in the guts of mammals. Okay. And here's a picture of a movie of swimming E. coli. Okay. And I like Berg's description of these. E. coli is a self-replicating object, only a micrometer in size, can swim 35 diameters a second, taste simple chemicals in its environment, and decide whether life is getting better or worse. Okay? Okay. And here we go. So, in spite of the overwhelming drag, E. coli are remarkably fast. So I compared Michael Phelps to an E. coli. So in the 2004 Olympics, Michael Phelps won the gold medal in the 100-meter butterfly in a time of 51.25, which is about 1 body length per second. An E. coli swim, in spite of the incredible odds, through this very viscous fluid at about 10 body lengths per second. So I awarded the gold medal to E. coli. Okay. Well, if you noticed, you saw this sort of wagging tail behind. E. coli actually swim by rotating a rigid helical flagella. using an electric motor. Okay? The thing spins, right? Most people would think that, that you see pictures of maybe sperm cells that wag a tail behind. Okay? But bacteria actually swim by rotating a propeller. Okay? And here's a nice comparison. In the 60s, Feynman offered this $1,000 prize to anyone who could build a working motor 1/64th of an inch on a side. And of course, this challenge was met, and here they are looking at it. Well, E. coli's flagellar motor is more like 1/640,000th of an inch on a side. And by volume, that's a million times smaller than this micromotor that won the Feynman challenge. Okay? How is it discovered? See, the flagella is so small, you can't really see under a light microscope whether it's wagging or rotating. So how do you actually figure out which is which? Well, this is what was done. E. coli were grown whose flagella binds to a certain protein. And they covered a slide with a certain protein and put a drop of water containing E. coli on the slide. And this is what they saw. Okay? So they had no idea whether it's wagging or spinning. And that's what you get. So we've actually caught a bacterium by the tail, and you can see the cell body spin. Okay? And actually, if you watch them a little bit, watch them for a minute, you'll see that they spin one way and then spin the other way. Okay? Okay? Well, that's a big part of their strategy for finding their way up favorable chemical gradients. What they do, so E. coli actually know probability theory. Okay? So what they do is they swim in a direction for about a second, and then they roll the dice. They tumble. They reverse their engines, and they sort of randomly tumble in the water. And we actually have an expert here who could add some more pieces to this puzzle. But very simply speaking, they roll the dice and choose a new direction and swim off for about a second. Roll the dice, swim in a new direction for about a second, right? And what they do is if they sense that they're going up a favorable chemical gradient, they just slightly delay the tumble. They slightly delay rolling the dice. And of course, why do they do this? Well, they can't just simply figure out that they're going up a good direction and keep going in that direction because they're subject to Brownian motion. And in a number of seconds, they get knocked off course, and they've completely forgotten which way they're going. Okay? So what they do is sort of a random walk with drift. Okay? And they take sort of this random walk, and on average, they wind up going in the direction that they want to. So that's how E. coli have evolved to control their motions. Okay? So they do this sort of random walk. And you can see these guys random walking through the field. Okay? Well, these are the guys that I study. These are bacteria that were just discovered in the mid-'80s. And they swim with no apparent moving parts. And this is a mystery that Jair and I have been looking at for about 20 years. Howard Berg brought these to our attention, and we've been trying to figure out how these things swim for 20 years. Okay? And you might think that, these are just some things that are you only find in very remote places. They're actually, by mass, the most common organism on Earth. Okay? They live out in the middle of the ocean, though, out in the ocean deserts, where people thought previously that there wasn't any life. Okay? And some water samples were taken, evidently, and more chlorophyll than expected was found there. And so they took samples, looked under a microscope, and the water was just teeming with these things. Okay? So these are very, very common organisms. Okay? And just real quick, since I guess I'm out of time. I'll show you how we think they swim. Okay, this is a very recent discovery. This is another bacterium that actually glides on surfaces, And what we discovered inside is that there's a cytoskeleton. Okay, so it kind of holds structure to the cell wall. But what we found is this thing actually rotates inside. Okay, and it's sort of this double helical structure, rigid structure, and it's driven by protein motors. And this organism actually uses that rotating cytoskeleton to propel itself across surfaces. Okay, and this is pretty well established. This is actually a movie of the thing moving. Okay, we attached proteins, right, that fluoresce. Okay, and we're able to actually make movies of this. Okay, and what we actually believe is that this is the mechanism that causes Synechococcus to move. Okay, so these guys that have no apparent moving parts. So we actually believe that there's a rotating helix inside of these. Synechococcus has essentially the same motor parts inside. And we've done some experiments that knock out swimming in both organisms. Okay, so same motor parts, and so we got the idea that maybe the same thing's going on inside of these mystery swimmers. And over on the right are actually better pictures. These are ones that these are pictures of our model from Scientific American earlier this summer. Okay, so and all done? Okay. Oh. I'll just leave you with this. Kind of creepy, but somebody right before my trip got a hold of me, and I arranged for him to get some cells. And he works at this visualization lab in MIT, and he has a way of amplifying motions. And so what he's going to try to do is look at these cells using high-speed photography and use this software to see if he could actually find the oscillations that we predict in our idea. So, okay. And I'll leave it at that.